when you see the word exosome in a vial, what goes through your mind as a scientist?
Well, for me, it's like, let me test it. I totally believe that the future of the medicine is vesicular. Now is the Wild West. Why to wait to someone do something if I can do something?
Yeah.
So I started to do the thing.
Do you think exosomes are a drug, or do you think the FDA needs to create a new category for exosomes?
Yes, they are not a drug. If you watch the drug description, it's not a drug, it's not biological, it's a totally a new category.
So you see a lot of manufacturers using bovine serum?
There are. And there are others. What they do is to put the cells in a starving.
They starve the cells?
Yes. That is a stress for the cells, so the information that they will put in change. Those are particles, not exosomes. That is just particles. And I become crazy when I see some certificate of analysis that say, okay, yes, we did the nanoparticle tracking analysis, and we have trillions of exosomes. And I say, what you are talking about? Show me that.
So, Dr. Mazzeo, you have been studying exosomes for 22 plus years. You've been a scientist for 30 years. And why is it important for doctors to know what's in the vial, in an exosome vial to be exact?
For a start, well, that is important because they want that the vial really have exosomes that have the potency to do the work that they need, that they want to do, yes, on the patient. That is in relation of to have their functionality, to have their potency, and really the integrity that they need, yes, to function, yes, to do their work on the patient. And the problem now is most of the physicians don't know right now. They can't trust of what is in the vial. So that is the main point here, is how the physician and in general, yes, person that want to use the vesicles that are in those vials really work, yes, in their skin or whatever way that they can use that. So that is the main point. What if the vial have what the companies say that the vial has? Yeah.
Okay. So when you see the word exosome in a vial, what goes through your mind as a scientist?
Well, for me, it's like, let me test it.
Okay.
Yes.
And as of now, there's really no one really a third party testing that is looking at all the vials and all of that.
Actually, you went to the point. The other main thing is to check if that product were tested, or the vesicles on that product were tested for a third party. Because, well, of course, when a production company from the manufacturing place, they in the production system, when they have when they purify the exosome, and after that they need to test that what they were producing were really vesicles. So they test that, and after that they present that test as a certificate of analysis. But that doesn't say nothing or have a lot of conflict of interest if not a third party, yes, tell them, okay, characterize your sample, and this is what you have there, and this is the integrity that your exosome has, and this is the quantity that you have in the sample, and the membrane is okay. And I will go further. I will say in relation of the functionality also. So to prove that what they have in the vial really, yes,
Okay.
work. Yeah.
Yeah. Yeah. We apologize about the banging. We got a
Yes.
comment
Yeah.
asking, yeah, for the banging. So I apologize of that. That it's very difficult to hear. But yes, give us 1 second. Yeah, there were when we were prepping for the video, we heard that, and she went upstairs to tell them that we were about to record and if they can keep it low. And I don't think they listened. So we apologize about that. Um, yeah, we got two different messages telling us that it's difficult to hear us. So I apologize again about that. Yes, yes. So you were talking about there's no standardizations right now in the industry, and what goes through your mind as an exosome scientist when you see a vial with the label exosomes, right? Yeah, we see that term being loosely, and I am obviously always trying to understand what's out there, what products, and I mean, I'm not a scientist at all, but I really try to understand the basic and the foundations, right? To then educate myself and understand and bring on scientists like yourself to that knows the science and what's really inside the bio scientifically, right? And the term exosomes, I see that it gets used very loosely. From what I've known, that every cell in our body secretes exosomes, so the source is very important. I've also seen like exosomes coming from plants, and so when it comes to an exosome vial, in your opinion, how much do you think is actually true what the claims are being made in the vial? Does that make sense what I asked?
Yes. What's exactly, yes.
Yeah.
How to know that what uh, the the company say that it is in the vial really is that. Yes, that you are. That is the thing that you are asking. Yeah. So that you have, yes. The thing is that the only way to know that is to ask question to the correct person, because sometimes the vendors uh, don't have all the all the information and all the knowledge. It's good also that the company educate, yes, to the people that start to sell, and also in relation of the regulation that we will talk a bit later. So the main point is that you can trust of that, educate you also as a physician, but also the only thing that you can trust in specifically that specific product real that you want to use, that you have in your hands. The only thing that you have and trust, this is the certificate of analysis. So but right now the companies are not given, as I say, what they are given, or what the information that they are given in the certificate of analysis most of the time is not sufficient, yes, to know what is real in the VL. And sometimes you don't have that information there. So
Yeah.
it's like you are just blind.
Yeah.
Um, and also the information that they give is from what they were tested, not as I say before, and I don't mind to repeat it because this is very important, that not for a third party. So that is like fundamental. And if you have the lucky to have some information in the certificate of analysis, you have to see not just one of the characterization process. What companies are showing now is their own analysis, the one that they do in their
In-house.
own laboratory, and most is the nanoparticle analysis. Yeah. So that is a nanoparticle analysis. In any place, say that is a nano vesicle analysis or
Yeah. So
nano
what does that
exosome analysis.
so for the average person, right, that doesn't really understand what that is, what is nanoparticle analysis? What is that? And
Yes.
why is it not accurate when a company talks and says, we have X amount of billions or trillions of exosomes, right? So like, what is that?
Yes. The nanoparticle tracking analysis. This is a general name for different machines that have different fundaments to measure particles in the nano size, yes, world. So we have different equipment that do that, and from different ways to analyze that. And what's those equipment have the capacity to tell us if what we have in the samples or the particles that whatever, that can be whatever thing, could be whatever thing, the breezes, all the thing, whatever. What we have there, the
Hey,
particles.
Dr. Mazzeo. So, the audience is pretty much saying that they cannot really understand, like, hear you because of the banging. And
Will go and ask with them.
yes,
You continue explaining.
yeah,
Yes.
yeah, I can kind of keep them
And
occupied, I would
I
say.
will back in a minute.
Yeah.
Yeah.
Okay. Yes.
Or-
Thank you. Sorry. Yeah. Sorry, guys. Yeah. Let's go. Did you tell them that they didn't listen to you the first time?
Like,
Okay. All right. Good.
but
So, all
this
right,
is
guys.
under construction. So yes,
Okay.
this happy that yes, I know the
Okay. Good. We're back. So, the only thing that I asked for you, Dr. Mazzeo, is just to speak a little bit louder, just so we can, kind of hear you, because
yeah,
I know that they were having some
perfect.
issues with
Yeah,
that. That's
that
the only thing I ask. And then, good. I was keeping the audience occupied. Okay.
that's good.
So,
Thank you.
yeah. Okay.
And thank you everybody. Yes.
Yeah. Thank you.
I'm so glad that all of you are here, and yes. And yeah, sorry, my apologies for this inconvenience. Yes, was no planned this way, but
Okay.
thank you for
So,
your patience.
yeah. So the question that we were in was essentially like, I was talking to them about kind of what we were going to cover about the certificate of analysis. I kind of prepped them on that we're going to go over a certificate of analysis, how to, like, what to look for, what questions to ask, what to vet, right? Like what questions to, how to vet a vendor, all that stuff, right? To protect obviously ourselves, the industry, and even the patients, right? Obviously, most importantly. So we know that the term exosomes
Yes,
gets you.
but I was sorry, Oscar.
No, go ahead. What were you going to say?
But I remember that I was telling, yes, the different things that they might say in the certificate of analysis before to go further on that. I was explaining the nanoparticle analysis, tracking analysis. Yes, that is name different kind of machine with different fundament to analyze particles, nanoparticles, yes, in the range of nano size. So they give information about whatever thing that you have there. Could be vesicles or whatever other thing. Yes, but
And in that sample, it can be literally any particle within that nano size.
yes, and
Correct?
they are using for different kind of science, also biomaterials, bio-nanomaterials. Yes, whatever material, from the cell, also that machine come from that part of science. Yes, for materials. So from there, that is the information. You have just the size of particles and the number of particles. But this is the first characterization to know, okay, if we have something here, whatever it is, which is the size of the things that we have in this vial and in this vial. And that is to say, is in the range of the exosomes or not, or we have bigger particles. Yes, that is the answer that kind of machine give us. But this is not sufficient. This is just, okay, we have something on the size of exosomes, for example. Yes. Or give you, okay, another answer that they give you is that If you ask after the purification, how much I enriched my this product, this sample, on
What do you mean? What do you mean by enrich?
enrich is to have more of that kind of thing. Yeah.
So they manipulate that in a way by adding certain things. How do they do that?
No.
How do you enrich something?
When I say enrich is when it's a way, maybe it's not the best word to say, but when you purify, yes, the from the media, the vesicles and the exosomes, we use a kind of filtration, yes, to separate the higher, the bigger particles and the smaller particles, and you want that sample keep in the range of the exosome size. So you have, what you do with that is in a way to in your sample you will have more vesicles that are in the range of the exosome is what you want to have, yes, in that product. So that's what I mean after the purification. Yes, it's not that you add things. Maybe I use wrong the word. Sorry, is yes, is how you purification. But when I say purification, people understand that, okay, I purify, so I have pure exosomes. And that is not the truth. For that reason, I'm also very cautious to use the word purification itself. Yes. But it's not also enrichment because you are not putting something there, something more. You just keep, yes, just take out what is bigger, what is smaller, and you keep in the your products on this on the range of the exosome size. But
Okay.
you don't have pure exosomes there. You have other vesicles that are in the same range, yes, of size as the exosome. Yes, right now there is not a safety process to exclusively obtain the exosome or exclusively purify the exosome. Yes.
Gotcha.
So
It is
that is one of the characterization that just tell you, okay, you have particles on the size of the exosome. So, and what you see on the certificates of analysis is that most of the time what they say is that, okay, you have, instead of particle, they change, I put exosome. This quantification and the size of all of those exosomes is this because the nanoparticle tracking analysis say that. And I say, no way. Those are not exosomes, they're just particles. You have to continue characterize that what is in the vial, that is through. That is the size. Okay, you have things that are in the size of the exosome. The other characterization is to say that have the morphology. So the other question to answer is those particles has the morphology of a vesicle, yes? And the second is that vesicle have the shape of an exosome. And that is the transmission electron microscopy that give you that information because you can see directly, yes, the
okay.
shape, yes, the
Yeah.
morphology. So that is the other part that tells you that you are in the that you have well something that it's a vesicle. And the third, the other, and after that, you have to ask yourself and ask the question, okay, I have something that is similar to a vesicle, have the size similar to an exosome. Now, this comes from the biogenesis of an exosome. And for that, you do all the markers that are exclusively the tetraspanines tell you more that, okay, this is a vesicle because all the tetraspanine are in most of the vesicles. But if you want to go further and say, but this is exclusively an exosome, you go for the biogenesis markers. So you label those vesicles that you see the integrity and the morphology and the transmission electron microscopy, and you say, okay, this vesicle that I see here have markers that come from the biogenesis of exosome, from the multivesicular body inside the cell, and those are unique for exosomes, and that characterize an exosome. Exosome, no cellular vesicle, exclusively an exosome, if you use the markers that are exclusively for the biogenesis of exosomes. And I can say that because I did my thesis on the biogenesis of exosomes. So I was studying the multivesicular bodies and how the exosomes are secreted. So that is fundamental, yes. And what is so in the certificates of analysis, you must see the size from the nanoparticle analysis. You have to see an image if it's scanned, or something that say that they tested that the integrity and the morphology for transmission electron microscopy. And also you have to see that they check if they have not only the tetraspanin, but also those molecules that come from the biogenesis of the exosomes.
So pretty much what I got from what you're saying is that if a company or a manufacturer is just counting exosomes or particles, then that's not. First of all, they're not even counting exosomes, right? So they're counting particles, right? And then in order to get an actual true exosome count characterizations to see what's in the vial, there's 4 different methods that you have to go through. So you said size, the imaging, morphology, and the biogenesis, right?
3,
So,
and I will, as you say 4, but are 3, but I will add another one that I really want to, and
okay.
also from the ISEF, we are start putting that inside the guidelines, is the functionality, that they are still functional.
That's
Because
right.
whatever process that they do during the manufacturing, that or whatever modification in a way that they do also in the origin and the source, that could in some way reduce the functionality. And I mean the functionality because we can see the vesicles from 2 sides. One is, okay, what is this vesicle? And the characteristic and the morphology of this vesicle, if an exosome or an ectosome or different kind of vesicle, yes? Big vesicles, big extracellular vesicles, or small, or all that thing. But the other way for the functionality is to see, yes, the vesicles and the exosome from the view of the cells that will capture them. And what we want doesn't matter how many, yes, vesicles we have there, or well, yes, mostly from the size. The main point is that those vesicles are going to do something, have still the potency and the functionality to do something on the capture cells. That is well, I would say that the main point. Yeah. So one part we have or not those vesicles in the vial, and the other question to answer is, they are functional, and the potency. How is the potency? Yeah, I don't know. And they are amazing tests that you can do. So that answer is not that, oh, well, we are not putting that information there because it's not possible to obtain that information. No way. In the lab is very easy. We do in the lab that test.
Yeah. Well, that makes
And
sense,
that's
right?
also, sorry, I want to add that. That is also different with what they show, which is the efficacy. That is another totally talk on the patient, yes, the after and before. But that's telling you, well, they have also more things in the sometimes more for the cosmetic, yes, and in the vial. So, yeah. check the potency and the functionality.
Okay. So if you're what I'm getting from what you're saying is that a manufacturer that should be providing in the COA the four different methods, but in your opinion, five different
The
methods.
minimum, the three characterization analysis. The nanoparticle tracking analysis, the transmission electron microscopy, and the biogenesis markers. That is fundamental. That is to say, okay, I have vesicles there. How they work, how the function, okay.
That's another.
But really, little by little, that will become part of the
Well, I mean, I think the functionality, not even on exosomes, on everything, right? Because you can have X amount of, exosomes, proteins, or whatever, but if they're not functional, then it's nothing. Nothing is going to happen, right?
Exactly. So the functionality is worth to have it. But minimum that you have really to ask to the company that provide those vials with vesicles to you, and to also, well, that is another thing, to prove that they are also what they say. If they say that they have exosomes, to really prove that they have exosomes is with the markers, a specific marker from the biogenesis, and this characterization is the minimum. The minimum. It's not just a nanoparticle, tracking analysis. That is just particles. And I become crazy when I see some certificate of analysis that say, okay, yes, we did the nanoparticle tracking analysis, and we have trillions of exosomes. And I say, what you are talking about?
Yeah.
Show me that you have that. Okay, show me that you have those trillion of exosomes in the vial, characterizing them, not just show me this nanoparticle analysis.
Yeah, and I've been hearing a lot too that the certificate of analysis can be manufactured. Like companies can make that. It's just a document, right? And
I repeat again. That's not the second question. First is you have all the characterization techniques and the certificates of analysis, and the other question that you must make is like that characterization was made in a third independent laboratory, not in your own laboratory, because of course that is something that you can from the is the bad sides of those characterization is that you can, from the scientific, well, I will say that later. The bad side of those techniques of characterization is that they can be easily modified and really show what you want to show. And those characterizations come from the scientific world, when really with the science, and we really want to not to see what we want to see. We want to see what or a study is meant to answer the question that us with scientists want to answer. But that is So for that reason, the more credible certificate of analysis and is when they show as minimum those three characterization, and that characterization come from an independent
third
party lab.
Yeah. So pretty much you're saying that the label and the certificate of analysis doesn't really say much. It tells the doctors and it tells us very little. That's what I'm getting from that, from what you're
yes, how most of the certificate of analysis that I read are like that. But if you have the certificates of analysis with the information that I give you, that's telling you a lot.
Yeah. Do you think I should pull up the certificate of analysis now, or do you want to wait off a little bit?
Because we wait. Yes, we will.
We wait. Okay. Okay. Just making sure, because I know we're talking about the certificate of analysis. And okay. So we know that, and we actually had a comment, and I would like to kind of transition a little bit towards the regulatory side of exosomes. So we all know that the FDA, they have not, there's no exosome product out there that has been FDA approved, right? And the only use is the cosmetic use, right? And so where do you see the future and what needs to happen in your opinion for, let's say, the FDA to kind of even look at this, right? And take this seriously, the exosome. So I'm sure standardization. Where do you see the market right now with that?
Yes. Well, if you let me, I want to go, yes, a bit back of your question, and to say that really, well, we can use, and most for human exosomes, also, how can I say that? If also, and cosmetic. So for the cosmetic people have to be very careful how they promote those
Are they
products.
market it?
Yes,
Yeah.
because those vesicles, or exosomes that they produce have to be just for, how is called this, uh,
Topical.
yes, and beautification, and just that, yes, and apply
What do you mean? I mean, what do you mean by beautification? Because I feel like beautification is
beauty.
broad and can mean a lot, right? Like, what
Yeah,
exactly do
but
you mean by?
just to beauty, to make your face more bright, or to, yes, and to but you can say that could, I don't know, improve some condition, or to do, because all the things make, for the FDA, is like, see that you are treating something, and with that they start to saying that you are using the exosome as a drug. Yeah.
Yeah, yeah,
That is the main, because can be used also for the cosmetics, for this kind of thing. It's just for beautification, just that.
gotcha.
If
Okay.
you use the exosome and cosmetics for other thing that is not beautification, that is no way. Yeah,
Hmm, yeah. Well, and the biggest thing is making claims online, right? Obviously, like you can't say that
I
exosomes,
agree.
help
But all the
cures with
you
this or
have to
treats with
change
those.
all the vocabulary,
Yeah,
and be very
which,
careful of that
yeah.
that vocabulary. And the
Okay.
other the other question for you, was,
About the standardization, where,
yeah.
what
So right now, yes, for the
it's the
for the
Wild West right now.
exactly. Uh, but it's normal, because this is a new topic, and it's not an easy one. So you really need to be educated on this and how this works, because it's very, well, not an easy yes, nanoscience also, and how the also the from the scientific world, also the knowledge, we are learning more and more, and doing more
Yeah.
studies on that. So the
Well, and then the translation from the laboratory out to the
exact,
commercial as well, which is kind of like that's what
that
I
is
see
the
where it gets lost, from the laboratory out. So
that is where things appear to have, because the standardization. So first, every lab to purify the exosomes, more or less the same way. But it's also totally variety in relation of the source also. So how you make that something, yes, standardized, if we have so many source there. The other is, yes, the batch to batch consistency. Yeah. The reproducibility. Yes. So those are all the things that we have to be patient in this field. Everything will come. This is totally new. This just jump from the science in the academia um, to the um, to the to the to the industry world. Yeah. So we have to go little by little, and that will come. So that is the way. That is the way that also little by little the FDA will see this in a more safety, yes, way,
yeah,
safety for
Of course.
for the patient, and that is the best part. But from that, I'm really working hard. I want to say, because you bring this at this point, that I'm really working hard to do something that is totally independent of whatever production, and to work with different laboratories around the world to make those standardizations. So what I'm working with different, just testing, they are not production. That is a rule. um, Testing labs, where what we are starting first is to analyze, yes, all the process, and um, in try to improve the standards that we have now, and to create one standard that
Yeah.
that the companies can follow, and after that is to present that to the FDA and say, well, we work on this hard, and this will bring with all the information that we collect from industry. Yes, not so for the from the science, that's from real product. Yes, that they are there. So this is information that we were collecting in this period of time, and we have this standardization. So I'm working hard on that, and I'm also invite people that are interested to participate, that have those kind of laboratories or knowledge, to join me in this. So we are working on that. We need to do
Okay.
that. So
Yeah.
someone have to do that. It's like now is everybody say, well, yes, it's the Far West, but what we are doing to solve that? I'm totally believe you. You know my
Yeah.
business card
Yeah.
and
So
my pillars. Yeah. So it's yes. I create this, and someone have to do something apart from what we are doing from the scientific part, but from the industry, and to make this, because, well, I say this and I didn't say, I put always here, because I totally believe that the future of the medicine is vesicular. So for that, because I'm totally in need of that, I put all my effort to really standardize that, and to make this in a way reproducible for the companies, and really work on specifically that, to make this possible for therapeutics, and possible for diagnostic, and possible for the drug delivery, which is all the potency that those vesicles.
okay, I see. So I want to ask you a question, and I put a poll right now, and right now it's
I love
50-50 right
that.
now.
Thank
And it's a very good question, because we all know that the FDA puts biologics, let's say, into two different buckets, right? You have 361, which is a tissue-based product, and then you also have a 351, which is considered a drug, and you have to go through all the the BLAs, the INDs, all that whole clinical phase products, right? It takes millions of dollars and a whole bunch of years. And so, do you think exosomes are a drug, or do you think the FDA needs to create a new category for exosomes, and why?
Totally. Yes, they need, and I'm totally on that, and working on that. I'm working to present all the evidence, and after that to go the FDA and say, well, you really need a new category for the exosomes. They need that. They are not a drug. If you watch the drug description, it's not a drug. It's not biological. This is an active agents that works in one specific way, which is, well, cell-to-cell communication. It's a totally a new category. So that must be there, and with a specific pursuit, a specific, everything is so specific for this nano vesicle wall. Yes. So attached to that category, all these specifications, how to produce and how to, yes, treat with this specific nano
Yeah.
wall vesicle.
Okay.
So of course, is need another category, but everything will come. We have to just be patient and not desperate. Everything is like it's coming. The one thing is that we have to do the thing the best way possible with what we have right now.
Yeah.
Not to do, yes.
You know, Dr. Mazzeo, I agree with you that things will come, right? Things will come. But I'm going to push back a little bit here. I also disagree with you where we have to wait, right? Because if we know how the FDA works,
No,
right?
no,
And if we
I
wait,
think I think we
if we
have to
wait,
wait,
they're gonna
but that doesn't mean that we have to wait. I'm not waiting.
okay. No, we
I'm
have to
not
push
waiting.
and we have to create these
Ex-
standardizations.
Exactly. I'm not waiting. It's not
Yeah,
what I say. Is that we have to be patient. Yes.
yeah. Patient and push them too.
Yes,
Yeah,
but I say no start to do what I say we have to be patient is that okay, to recognize what we have right now, do the best that with what we have right now, and do things, of course, because we need it's like when I started with I started to build this that I'm breathing right now and to create the standard, yes, for the exosomes, and that is that I was thinking someone have to do something because now is the Wild West. But if I stay there, that is just wait. I say someone have to do something. But when I start to thinking, I say, come on, why to wait to someone do something if I can do something?
yeah. Of
So I started to
course.
do the thing. I know what we have right now and the possibilities that we have right now in this, and that will be. I totally believe on this, and that
Yeah.
made it.
So, Dr. Mazzeo, for the people that are just watching, and they just heard of who you are, and because you're showing the little card, but we can't read it, right? So, since you're talking about it, we're on that topic. Can you go over and explain a little bit, like what are you showing and what are you working on?
Yes, yes, I will explain, but I would like to also to explain this in an extent, maybe in another podcast or maybe to yes, for people
Yeah,
that.
that needs probably a separate podcast to literally get into
Yes,
the
totally.
details.
I
But
will I will
what are you
tell you
working
that
on, and what?
yes, because I know that people want to know more in relation of yes, what is in the vial and how they
Correct, but you've been showing the
yeah.
card, and so like what is that, right? That's why I want to bring it up.
Yes, this is what I'm creating is something that is called Safe EV, which is the S is for standards, the A is for assessment, the F is for exosomes EV, and it's a E with a tick. So this safe is we are now getting agreement with A9 laboratories around the world, and we are working together in all the characterization and in all the really working on that. It's not just giving guidelines and all that. It's just to take all that information and really do something to improve this field and to really put the standards there and as I say, and after that to talk with the FDA and really start to do things in this field. This is going to be a certification mark also internationally. So we are going to start certify the process. So we are going to start for those companies that really want to work and do the correct thing at the time that this field is improving. We can help to improve also their production, yes, in the proper way. And also together, yes, put all the information there to after that improve also what the FDA are doing with this in this field with extracellular vesicles and also educate them in some way and with the data that we have. So that is what and also the possibility also to certify them from the cosmetics also the formulation and see if that modify in some way the vesicles that are in the formulation, all the thing we can test. And also for us, it's also information that we can keep to also show later for those vesicles to be approved, yes, for everybody, and also in on the trials and all this. So
Yeah. So you've obviously you tested several exosome products in the lab. You've characterized them. You've done a lot of work, right, with exosomes products. what are the most common like safety and quality problems that you see across exosome products?
Yes. Thank you for that question also. You always go with a good question.
Those are all the questions that I ask myself, so I make sure to write them
Yeah.
down and ask them.
That's,
Ask
that's
you.
amazing, and it's something that people don't talk too much. So that is
Well,
also,
we have to be objective, right? I
yeah,
think especially in science, like I want to know. I like to know the facts, right? And if the facts are the facts, you can't fight that, right? Like that's the fact, right? So like I try to obviously be very objective when it comes to like understanding,
yeah, and
especially if we're really trying to
to bring
help patients, right? So
exactly, and bring the truth here. So the main point also relates with the production and the manufacturing process. That comes from the source of the cells. We start from there. Well, there are different sources that companies are using now, more from the stem cell world. But apart from that, that's also the cells itself is also another, what can I say, another postcard maybe to talk exclusively in relation of the source of different of the vesicles. But when I name here whatever the source, I say the source is very important in relation of what those vesicles will do in the patient. That is first, but exclusively talking about the safety part, know what they will do, yes, therapeutically or in this way, is more in relation of the safety. For that, you have the manufacturer, and that is a question that you can do to the manufacturers of your product, yes, or the vendors of those products. The vendors maybe don't know, but he can figure out how to find that those answers. Is how in which media the cells are growing. That is first question. In which media? Because we in the lab, for example, when we started, we use a media because we are just, I don't know, apply that to other cells, yes, or we are going to treat cells or to treat mice, but the safety for humans, you have to ask in which media they grow those cells, and also how they maintain, yes, those cells in the culture in relation of the pressure of oxygen, the pressure of CO, and all the things. And they are using uh, which serum they are using. The other day, I was talking with one person that she is very related with different manufacturing companies, and she started to tell me that laboratories are using um, um, bovine
bovine.
serum. And I say, how that can be possible?
So you see a lot of manufacturers using bovine serum.
That is not the one that I was connected and checking, but there are, and there are others that they decide not to use the serum, and what they do is to put the cells in a starving. So
They
that,
they starve the cells.
yes, without food.
What does that mean for like a non-scientist, and
Well,
why is that bad for the exosomes?
that is a stress for the cell. So the information
Mm,
that they will put in change. It's
interesting.
different. One cell that is growing good with a nice environment, and a cell that is in a stressful place that is starving, and that some way we do that also to I remember when I was in the lab to increase in some way that increase the production, but change in a way what the information that those exosomes most exclusively because come inside the cell, yes, the information that's from inside the cells go to other cells. So and there are other companies. This is expensive, but this the most way to do is to that they produce their own own media, yes, and sometimes they come from their own source of those cells, yes. So those stem cells. So from where they extract the stem cells, they use, yes, from where they extract the cells to grow those cells in the same environment that they were, yes, before, when they purify and when they take those stem cells from the tissue. So that is yes the best way to go. And
Okay.
that makes, yes, less problematics in relation of safety. Another point is there are some companies that don't purify and they just start mostly in cosmetics. This not so regenerative or in therapeutic. I think, but yes, they use the total medium, yes, where the cells are growing. No, but before to go that, I say how they grow, yes, in relation of the cell before to go to the medium. How many passages, if you are producing exosome from cells that are from because that is the best potency for the cells and the best functionality, the ones that come from the stem cells. But the stem cells is one specific period, yes, of those stem cells. If that exceed that period, they start to differentiate and they start to become other kind of cells. So to maintain that phase, you can make, yes, hundreds of passages and grow, and almost indefinitely those cells, because you are going to have another totally, thing growing, yes, there. So very low passages. So another question to ask the production companies: how many passages you did on this on
How
your.
many passages?
Yes, exactly.
Okay.
Exactly. So that is the other. And the other is you purify, really. That is for cosmetics, because
Yeah.
I know that
And
products that just sell the media where the cell were growing. And that is also.
they're just selling that? Wow. Um, well,
Yes, you have a question, yeah.
yeah, I forgot it because
Well,
I forgot the question, but it'll come to me. I'll ask
okay.
you.
So when it's come, let me know. Raise your hand. And the other is well, the media, and how the other part also for safety is the condition and how they purify, as I named before, that.
Okay, I remember the question. So the passages. So you said that how many passages the manufacturer is doing. So like, what is the optimal passages, right?
Oh,
Is there a cutoff? Like, what? Yeah, can you go over that a little bit more?
yes, that is very, very low, 4 or 5. Yeah.
4 or 5 is like okay.
Yes.
Okay.
Yes, after that. And also you had to genotype the cells and check if they are still in that phase of the stem cell. So the science had lots of, how can I say, lots of procedures that you can use to really test the safety. Yes, and the manufacturer, we do in our lab also to publish. We have that. If not, when we are going to publish or project, what's happened is the reviewer asks us for that thing, and we must, if we want or or a study, we publish it. So we have that. And also it's good that also the companies outside also test that as we test when we want to put the truth there. Yeah. And also in relation of to put we when well when scientists have a project related with the vesicles and exclusively exosomes, and we want to name exosomes in our manuscript. When we submit the manuscript, in that manuscript we must show all the characterization, the three kind of characterization, just to put the word exosome on that manuscript. If not, the manuscript and the submission is rejected for the journal. But now you have lots of products without that characterization that put exosomes in the packaging with
Okay.
characterization.
So that's one thing. So like also one question is like, where do products lose integrity? Like, is it in the storage? Is it in like the whole supply chain before they even reach the patient? Like, where do you often see that products lose its integrity and its quality, I would say?
Yes, there are different parts where that happens. Let me start because we were talking about the yes the
Passages?
no, the purification process. So in that step, for example, there are different ways to purify, yes, the exosome. But in those,
Mazzeo, sorry to cut you off. Is purified the same as isolation?
yes,
Okay.
yes.
Okay.
How is used in this context? Yes, yes, isolation or purification. Um, uh, so in that there are different kind of process. And still, there are companies that use ultracentrifugation. I was using time ago yes, 20 years ago when I started to study the vesicles. And most of the time, I had to change at that time as pursued when I were with ultracentrifugation. So that is a technique that is good for concentrate, yeah, for concentrate the vesicles. Yes, they separate and concentrate the vesicles. There are different kind of centrifugation process. But most of the time during that process, the vesicles aggregate. So when they aggregate, if you don't do another process to disgregate the exosomes. I was doing that in the lab. Disgregate the exosomes. They will reduce totally the potency. Yes. But also, if you add to the ultracentrifugation, if you add a second step when you separate and you disgregate the vesicles that are all together, stick it together, you lose also their integrity and you lose also potency. So for that reason, now what most of the production companies are using the size exclusion or the filtration, the tangential filtration when they choose just by size. But I said that is they purify, but they are not take the exosome exclusively because it's by size. And there are other vesicles that are not. I will say this in this way. There are vesicles that are negative for the markers, biogenesis markers of exosomes. That means no exosomes, yes, that have the same size of the exosome. Yeah. You have exosomes there with positive biogenesis marker. That is what say is the flag that say I'm an exosome. Yes, this is the exosome. And there are other vesicles, same size, yes, and more. The structure is another talk, but the same size that are negative for those, biogenesis marker because have another biogenesis. Yes, they come from another part of the cell, not from the exosome count. So that is also, as I say, the purification step is something that is good to know how they do because that also will reduce the integrity. And the other part is that in relation of most in cosmetics that they use a lot of formulation is the formulation part. So it is what you have in the formulation. Yes, because most of the components in the formulation. They break the membrane or dissolve the membrane of the vesicles, not just the exosome. The membrane of the exosome is very strong and could survive outside the body for long. So and some cycles when you freeze them and that. So, but when they are just maybe in the blood serum or in an environment that is good for them to survive, if you keep them or add to them lots of formulation and other components that are not typical environment for those vesicles, of course, those vesicles are going to dissolve and also the potency will reduce. And another part is in relation that I started to talk and the scientists start to talk. I don't know if to bring here because I'm also confusion on this side. That is the corona of the vesicle.
Corona.
The corona also, if you make, I just want to say, and I can stand that in another podcast if the people want.
You can briefly talk about what the corona is and what.
So that if you, that could change when you add component to the formulation and also for the lyophilization, and that is another topic, and all the things that change the behavior, how the exosome will be taken for the receptor cells and how they will behave. Doesn't change the characteristic, the cargo and the real intention and the functionality of that exosome. That's not, but yes, the behavior.
So the definition, just because I had to look up what a corona is for those that don't know what a corona, what it says. The extracellular vesicle corona is a dynamic layer of biomolecules that spontaneously adsorbs onto the surfaces of EVs like exosomes when they enter biological fluids such as blood plasma.
So imagine when the cell secretes the exosome, they inside the cell they are called intraluminal vesicles. Yes, and they are formed in the multivesicular body inside the cell. This is the cell. This is the multivesicular body with little intraluminal vesicle inside. When that fusion with the cell, with the membrane of the cell, the multivesicular body open, like this, and secrete all the intraluminal vesicle as exosome outside the cell. In that moment, if that cell is a cell that comes from the blood, for example, in that moment, lots of different biomolecules that are in the blood start to attach, to the surface of the vesicle, and that make the vesicle flow, and that also help in some way to be seen from the specific cell that had to be seen that those vesicles, and for the vesicle to do the work, and in this case exclusively exosome, because they are the functional part of the extracellular vesicles, and they are the one that have the capacity to start all the process of signaling and also reprogramming the receptor cell.
Okay, awesome. So I would like to transition. Is there anything else that you wanted to add there, Dr. Mazzeo, to that?
Yes. If someone wants to know more on that, they can contact me or maybe to tell us if they want on a specific.
So I can go before we transition over to the vetting your vendors, because I have a couple of questions there, like what questions to ask and all that. There were a couple of questions that they were going in, but they were being answered, and I think it would be good to bring them up. So one question would be, would you consider a passage of 4 to 7 acceptable for, say, HEC 293, HEK 293?
Well, that is another mention. The main point is that this is another thing that you have for the HEC cell. It's a cell line. So the main point is that it's not something safety, to use also a cell line to go to produce in the lab. I was using Turcats and different cell lines, but the main point is what I was talking is for primary cells, and most of primary cells, those that come, that are like stem cells more, that are indifferentiated, and to kill all the, how can I say, all the capacity of the stem cells, and all that capacity to keep all that capacity of the stem cells in the vesicles that the stem cell will secrete. They have to maintain this stem cell category, and that is not if you expand them too much. That they start to differentiate in another kind of cell. That is thing, and also using cell line to produce vesicles that human will reuse. That is no way, because the cell line, most of them are in some way they are transformed in some way, so that is not safety. That is not safe. So no, if the company tell you that they are using cell line, no way. This is so run away. And the other is what I wanted to say. There are other companies that are talking to start, and this is crazy, that they say that they want to immortalize the cell, so modify the DNA, immortalize the cell for them to start to grow, and that is also no way, because of course, if you modify the cell, you are modifying what they are secreted. So I will accept that if they show me a panel with all the study, all the omics, or all the characteristic of those cells that come from the immortalized cell, with a panel of a naive cell, all the vesicles, the panel of the vesicles. If they are identical, that is not. I would say, well, okay, go for it, but it's not. You check everything.
Okay. Can you mention if there are any risk with exosomes, even if they are properly produced? Like, is there any risk, right, with exosomes, even if they're properly produced?
I right now, my most trusted, because all the information that come from all the research that we have right now, the most trusted are the ones that come from the neonatal tissue. Why? Because they were not in contact yet with all the environment. Yes, that neonatal is totally new tissue. They are not from an adult that also have their own, I don't know, style of life, or also that adult environment. So those are the most trustful ones that to really,
I was going to say, but then obviously making sure that the donors are properly vetted, they're properly screened, right? Obviously making sure that
is something that, yes, I don't say that because I assume but that is something that you must see. Yeah, that is something, no way. In the certificate of analysis, in the paper, that they do all that research. That is a part of the vesicles. That is a good procedure. Yeah.
That's one of the procedures.
Exactly.
The must. Okay.
Exactly. That is for that reason. I it's something that I assume totally that they do that.
Yeah. Well, I mean, the way that I've seen it, I mean, we can assume, but I'm sure people are still cutting corners, right? Collecting tissues from people that are not following the rules, right? So
exactly. Yes.
I wonder how that should that be in the certificate of analysis too, like the screened, or for the patient as well? Like for example, like the endotoxins, all the donor information that they got screened for certain diseases and all of that.
Yes, everything from the patient.
Okay.
It's like it's whatever tissue is how you when you start working in the laboratory with a specific tissue that is a part of the vesicles, that is something that you must. Yes.
Okay.
Do. And the other thing also I want to add here is we have also the possibility, and that is part of the characterization also to add, is the impurities. You can test impurities. Yes, you can test if there are debris from the cell, for example, that are there. That is, they are totally negative in relation of different markers, and you can really with different markers that would really put a flag, yes, on those debris and telling you, okay, the sample have lots of impurities and also the kind of that come from directly from the cell, but doesn't have to do with vesicles exclusively. It's just debris of the cell or whatever, or and also and more if they are using bovine, yes, serum, you can detect that also. You can detect.
Why is you mentioned that? Why is using bovine serum a no-go when
Oh,
producing that?
well, this different things. That because it's another, yes, it's another species. Yes, it's not from human.
Okay, that makes sense.
And the other thing is also
So if it why are companies using it then?
what to feed because it's
Mm,
it's, a,
interesting.
it's cheaper. It's more difficult to find, yes, human serum or that or
So is so
another kind of. As I say, the best way is to from the place that you subtract the cells, yes, use them the main environment to grow the cell. But other thing important is that I suppose that those companies that use this the bovine also delete that serum from the vesicles that come from the cow.
what do you mean by delete? Like
Well, yes, you have to purify them and take all the vesicles that come from the cow out
mm,
because they have also, yes, vesicles.
wow,
If you don't treat first that
yeah,
serum, you are mixing all the vesicles with the human that come from the cell.
hmm, okay,
So what we do in the lab, we use because we treat just cells, not human or mice. And at level, what we do is to buy serum that were, yes, without any vesicle that come from other. So the serum, but also is with some, yes, waste, yeah. But it's depleted. It's depleted of other vesicles that come from other, yes, from the serum and not from yourself that you are growing to obtain your own, yes,
yeah,
vesicle.
okay. So let's transition over to vetting the vendor so we can start with that as well. Let's say for example, I am a physician and I've been getting a lot of asks from my patients saying, "Hey, I've been hearing a lot about exosomes and what do you know about them?" Right? And so where should a physician start and what my question is what questions should a physician ask vendors first? Like what is the first thing that they need to ask?
Well, first, to know the necessity of the client, yes,
Okay.
for what
Of
for
the patient, the patient,
the
the like
patient.
why, what is okay. So like what is being treated, I would say, or what
Yeah,
okay. So
why do you want to, or if it's the physician that offer, yes, that to the patient is, well, okay, what he offer and why to treat what, because also that depend of the 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 um, the proper vesicles or the yes the product, yes, that for what they will treat that patient. What I mean is that, well, from which source they will come, yes, in relation of functionality. So that is the first. the other is what we were talking in relation well of the safety, yes, that they can take care of. If it's the patient that come with that and say, oh yes, I want to treat this with exosome, I say, okay, first, what you know about exosome, and start to educate. That everything will start with education, yeah. And after that, to explain that, okay, right now the possibilities that we have also right now, because whatever the physician do on the patient, the responsibility go to the physician, yeah,
physician.
yeah. So, and this is very, now the regulation is very tricky. So that also had to be explained to the patient. Yes, the possibility that the physician had to work on that. And after that, explain, yes, what we told here. They say, okay, sometimes the patient the patient bring directly the sample. I say, oh yes, I want to apply that. Well, we know work with this sample that yes, I found, and this is from my friend that she found for whatever place and this. And and say, okay, sit and talk. Which is the source? Okay, you have a certificate of analysis of this. What is saying? And if not, and the patient is say, okay, the physician also can, if they have the possibility to test that product. Yeah, the physician can test, can do the thing that maybe the company didn't. It's like to, okay, I'm working also. I'm a physician, have this clinic, and I'm working. I do exosome applications. So I'm working also with a third party laboratory that tests the exosomes that come to my clinic. So the physician could have the possibility to test the products that comes to his clinic. Yeah. To test themselves and say to the patient, okay, I see here that there is nothing that can help you because I characterize them and there is nothing like exosome. I can treat you with another thing or a pure exosome, or we can find together really a source of exosome that is good for what you want.
Interesting. So when you mean the source, right, it means like, where is that exosome coming from? Does the tissue of, because what I learned is that almost every cell in our body secretes exosome. So like, and you said
the same.
Yeah, and you said like the neonatal, you said, right? So for example, I'm guessing what amniotic fluid has exosomes, Morton's jelly has exosomes, umbilical cord has exosomes, tissues has exosomes. I mean, is that accurate? Like, so like, all depending on that specific source of where it's coming, even in the whole placenta, that those exosomes differ on the functionality and what they do.
Yes, the cargo is like, I don't know, yes. For that reason, the stem cells are the best source because it's more wide. Yes, because they were not differentiated them yet.
Gotcha.
Yes?
Okay.
So if that comes from the stem cell from the adipose tissue or these kind of things, they are more
Like autologous, you mean, right? Like autologous adipose tissue from an adult.
autologous or no. I'm talking just the cell. You can,
Okay.
autologous is when you apply to yourself or other people. Yes, yourself is autologous.
Yeah.
But you can have some cells that are and start to produce exosome for other people, not for yourself, that come from a tissue like adipose tissue. Yeah. But this is not autologous, it's heterologous. So the point is that change in a way, or other kind of from other kind of specialized tissue. So that is more, yes, it's like a pear tree give you pear, yes, can give you apple. Yeah, this is the same, but they are fruit. Yeah. So they are exosomes because they have, for that reason, we can characterize them independently of the function. Yes, we can characterize them and say, yes, this is an exosome. What they do depend of the origin. Yeah, they are different in what they do, but they are the morphology, the size, the biogenesis marker are all the same for from whatever source, from whatever part of the body. They are all the same, and on the things that characterize them have come from the biogenesis because they are producing in every cell the same way. And also you have to ask, this cell have multivesicular bodies because that is the compartment inside the cell that produce them. Is that like, for example, not the human, but some people confuse and they start to talk about those from bacteria. Yes, those vesicles that come from bacteria. The bacteria doesn't have multivesicular bodies, so they don't have exosomes. Whatever company that comes and say, "Oh, I have exosomes from bacteria," no way, impossible to have exosomes. Yes, to produce exosomes that come from the membrane of the bacteria. Yeah, and have they own? I don't say that it's bad. They scientifically prove that they have the function. Yes, they work. But so no, they don't have the characteristic, yes, and the functionality of an exosome. Reprogramming the cell, all the things. It's different, yes.
Gotcha.
That is for other things. So if a patient come to you with exosomes that come from a neonatal and everything is good and producing good, and other that come from bacteria, they say, well, those are different things.
Yeah. Okay. Okay. So let's go over through a certificate of analysis, walk us through it. I'm going to be sharing our screen. So for those of you that are watching, you will be able to see that and kind of follow along with us. Let me just share this real quick and then we'll kind of go over and then let's see, share. Okay. So this is a certificate of analysis.
Yes, I see. Oh, no. Yes.
Can you see it, Dr. Mazzeo?
Mm-hmm.
Okay. So what we did, obviously, the point of this is not to
No,
it's not to shit on a company or talk bad about a company, excuse my French, but it's just more of like just educational purposes. And I also want to learn what I'm reading here, right? So please, Dr. Mazzeo, go ahead.
yes. Let's start with this because this product, they say, okay, so Wharton, the lots, everything.
It comes from Wharton's jelly.
Jelly, that is okay. I hope with Wharton jelly is amazingly to grow those cells in the to take from the Wharton jelly also where the cells are growing and make a medium with that and grow the proper cells in that Wharton jelly medium. That is so rich for the cell and you don't have to worry about that is formulation, filtered solution containing, okay, they have, okay, that is to, and that's it, exosome count. Exosome count, how they count. We'll start with here.
Okay. This one right here?
Yeah. I see that and say, I know that is not that way. No way. That is what I say. Those are particles, not exosomes. Because there is right now there is any technique that could separate safety. Yes, in the lab we can precipitate with the exclusively the exosome with how it's called this with magnetic beads, but that is in the exosome attached. So that is not safety. So that is particle. Okay, and what I read here is not exosome counts. What I read here is particle count.
Yeah. So do you recommend that like the COA should have particle count instead of exosome count? Is that like a little bit more precise and accurate?
Exactly. And if the company, let me check the rest. The size. Okay, that is not the size of it's the size. The most is what this machine give you is a mean is the media, yes, of all the vesicles. It's a peak, and the media is in the part of the peak.
I know. I think you sent me a picture. Do you want me to show that picture?
Yes, later.
Okay.
Yeah, I will explain that. But this is okay. This is the size. The majority of the vesicle have that size approximately. Sterility is sterilize. Okay, then the toxin, RNA. Well, I want to know how they measure that concentration, protein concentration. Well, this sometimes they still of give the numbers, they give the concentration. The way to really count the vesicles is for the particle count, not the protein. If they show you the protein concentration in microgram milliliters, that is you can differentiate for a lot of impurities. So you must have in the vial, the counting, the billion per milliliter, please. The counting, yes. Not that is more related with impurities. Yeah. Yes, they do the purity here. They say measurement, nanosite, purity, identity, the purity with the nanosite. I will want to know how they do that. Protein, one measure it. Okay, that is the impurity. They read the impurity. Okay. So this is not giving me, it's just, okay, they have a good amount of particles. Yeah, and it's sterile, they are particles. Now, what I need to add to here is the markers, the biogenesis marker, yes, and that also can be not directly. You can do directly on the transmission electron microscopy, but not a direct account. But you can count you can count that marker, and you can estimate how much really vesicles with the biogenesis marker you have in your sample, the proportion. And the other part is yes, Chaitanya say how they measure purity. Yeah, that was my question, Chaitanya. Yeah, the impurity. You use some markers and you go for the protein part. Yes. Well, you also, as I say, you test that with markers for specifically for that mark the cell debris or something that comes from the cell directly and it's not vesicle. Yeah. So the other part that I say is that is just telling me that they have particles. Yes. And the other and to what they need to add here is, as I say, the markers, the impurity measured as the markers that are not vesicle markers, that are cell markers. And the other way is to, of course, the integrity. For the integrity is to show that they did the transmission electron microscopy. And add the functionality and the potency, but that is extra. But at least that. Yes. What okay.
I zoomed out a little bit just to kind of see.
So description of the test should have the description of the test. When it was tested, who tested, and look, of course. Yes, Frederick, thank you for that. Yes, you have to know, yes. As all the information, which in there they have to say the source is there, that is good, but how many passages is good to have there. After that is how they grow the cells. That is great to have there. Maybe the patient will not know about all the things, but I hope the physician will educate himself to know if something like this, and also after this talk, yes, to know when they say if some certificate of analysis with all the description appear, and they say no, we use fetal bovine to grow the cell, and say alarm there. So that is. Yes, the main point. So this certificate of analysis for me, it's just, okay, you have a good amount of particle. What is in the vial? I don't know. That come from the Wharton jelly.
Yeah.
But okay. I don't know. Is it sterile? Yeah, okay. But
so I have a question. So like if I'm a doctor, right? How do I know that how do I check, or how do I know that okay, knowing what to look for in a COA is one thing, right? Like understanding all of that, right? Which is what you're doing, right? Then that's one thing. The second thing is, how do I know that what they're saying is true? How do I verify that?
For that reason, in that certificate of analysis have to be, yes,
A
a
third
no.
party, what you were saying, a third party?
Exactly. Exactly. If there is the stamp, because of course it's good if the third party give a kind of a stamp or something that say, we check it. Let's check it and it's okay. That is how you really rely on what this is saying. If the company tests in a third party, yes,
Gotcha.
laboratory, and it's totally independent with whatever other manufacturing or whatever, it's totally independent. They test, characterize the vesicles, the product, yes, the product, and say, yes, you have good, and this is all the characterization, and you can see here your amazing vesicle with all the structure, the typical cup shape of the vesicle of the exosome, and also the markers that come from the biogenesis. So you have exosomes there, maybe mixed with other vesicles of the same size, as I would say, but you have that. So that is amazing for your patient. You have everything there. Yeah.
Okay. Cool. Are you still have more comments on this COA, or do you want me to go over to the other graph?
There are some questions. Should have the description of the test. What do you mean by which test? When
Well,
is what tested? Who tested? Yeah. Oh, yeah. That would be amazing. Of course. And the law. Oh, yes. Thank you.
the lot number.
You are bringing an amazing. Yeah. Thank you. Thank you, Frederick. Yes, the lot, because yeah, you had this. This is the description. This is so important. Thank you to bring that, because I was forgot. This is all the description for one lot. Yeah. So you must put the lot number. I don't know. Is here a lot number?
I don't think so. I don't see the lot order number. Well, right here, lot number.
Okay. That is good. Okay. That is. So that is totally important. And for the patient to understand that this is that lot. From another, they have a totally different measurements. They have to measure all again. Yeah. That is the case.
Okay, cool. Yeah, Frederick said that you can request the raw data of the results. And they have the reference number from a third party. You can call the company and verify yourselves. Okay, cool.
Yeah.
Good to know that.
And not all give the raw data, but at least to have the last data that shows that, okay, that you did the thing. Yeah, you did the characterization properly, and at least you have the markers positive, good. You have the company doesn't have to give which marker they use, but I think that they did that. Yeah. It's good that they say, okay, we use these markers and this marker that, apart from the tetraspanin, we use those markers that's exclusively from the biogenesis. Yeah.
Okay, cool. Yeah, another comment was like, what do they mean by long-term storage? Do they mean 12 months, 6 months, 24 months, 36 months?
No, well, that depends. That depends of the really. That depends of the cells that you are using, because there are different. The cells have different timing, and they grow more slow and they grow more faster. But what I mean is how many when the plate is over, or the how is this the batch
Batch number, the lot number, sample.
no. When they when they grow, yes,
The medium.
the cell, no. They when they become confluents, they have to the cell don't have a space in there to continue growing, so they have to put the cell in another container. Yeah, that is one passage.
I see.
And you wait until those cells grow. This timing depends on the cell. As I said, some cells grow lower, faster, but you have to know your cells. Yes. the cells that you are growing in your laboratory. So when they we call confluence, yes, that they more or less cover the 80, 60, depend of the cell also, percent of the surface. Yeah, you have to put it in another container because that is overgrow. They start to die because yes, they grow overpopulation of cells. So those passages to one to another have to be just 4 or 5. Some, no, 7. But that I mean, the time that it takes is specifically to do those passages depend exclusively of the type of cells, because I say there are cells that grow faster and lower, but the number that you do the passages, and it's not just that. I say that, but in reality, what give you, for that reason, you have to, the production, the company that produce the cell have to really know very in deep the product, yes, and how they produce them. They have to know very deep the cells that produce. One test, instead of go for the passages, is to genotype the cell. And no, you maybe have to have that not all the time, just if you continue using the same type of cell, you can phenotype and say, okay, with how many passages phenotype in there, they lose this stem cell characteristic, and you can phenotype and know that. Yes, and after that you say, okay, this cell tends to have this start to differentiate in this number of passages. Okay, we are not going to approach this number, and I know because I phenotype, and I know exclusively in which number of passages this cell start to differentiate. Okay. And also depend on the media. You have to maintain all the media, all the serum, everything in place. Yes, this is another.
So, Dr. Mazzeo, quick. One thing that let's go back. We had one comment. She said, but did it mention what isolation and purification techniques that they used in the COA? Did they mention it? Is that important to include in the COA?
The way it is in the certificate of analysis is good to have as much information as we can. That will be amazing if the company say how the process of purification. That would be amazing to have. Maybe not compulsory, but at least to know how those cells were purified. Most of the time, it's tangential filtration.
Well, here it says purity and identity was determined by nanosite. Is that?
No, that is not. That is something that I don't know.
I mean, they're trying to answer the question, but it doesn't answer the question.
And also, it's the nanosite LM10. This is very old one. I was using that in 2008. Now is the NS300 or the Pro that also have an AI incorporated there.
Wow.
Yeah.
AI is everywhere.
Everywhere, also in the nanosite.
Okay.
So no, they don't say, they say filter more and the more up there around here. They will, yes, formulation, filter, solution. No, they don't say. So it's good to have that. That would be amazing. More information on how they, yes, maybe not something compulsory, but maybe I missed it. Oh, there was a question and that got from where?
No, it was the question that I asked. Maybe I missed it, but if mentioning the isolation purification techniques they use in the CUI, is that important? You said as much as information possible is good.
Yes, that would be, because more and more technique in relation of purification are coming, and different wise with different fundamentals. So there are some process that maybe that also there are some process that take off in the process of purification. They separate the corona from the vesicle. Yes. And sometimes it depend how you are going. This is not bad, not good. It depends later how you are going to use your those vesicles. Yes, if that is important for you or not. You have to know very well your cells, your process, and first of all, ask yourself for why you are producing those exosomes, and that is all the. And in relation of that, you start to build, yes, every step from the back. Yes, I want this kind of vesicles. Okay, which kind of purification process I need to have this specific? Which cells, which source, which cells we have to have all these steps. Yes,
Interesting.
knowing very well your product, and also, well, if you can do all the omics on your vesicles and your exosomes, will be great. That's for the company to know all the amazing capacity, yes, for all the components. That's the proteomic, the genomic, the lipidomic, yes, all the
You
vesicles.
know what would be probably a good idea for you to create a perfect COA, what it should look
Very
like.
good. Okay, yes.
That's actually, and give a sample
Yeah.
of like, and that should be probably a good
Yeah,
one.
yeah, I will do that. That would be
Wow,
fantastic.
that's a
Yeah.
good. I'll write it down so I don't
Yeah,
forget.
that would be fantastic. We can produce that. And here we have this also certificate. Well, this comes from a book from a company. Well, it's a kind of certificate of analysis.
So just to move the audience, so you're going over now what's on the screen, right? What we're sharing. Do you need me to zoom in a little bit more, or is this good?
A bit, but I know more or less. Yes. What I was talking is in this histogram. This histogram come also for the nanosite. Is one of the NTA of the nanoparticle tracking analysis. As I say, the nanoparticle tracking analysis is an analysis that cover different kind of equipment. Yes, that do that nano tracking analysis. Yeah. So there is no one that called NTA or nanotracking, because some people come. What I say that because some people come to me and say, "Oh yes, no, how you measure, how you characterize?" I say, "Oh, I use nanotracking." I say, "Yes, yes, but which one? NTA, nanotracking? No, but which equipment?" Nanotracking is the general, yes, indication for this particular analysis. Yes, but there are different ones. One is this that we present here. This is the nanosite. That measure, well, the fundament is that you have the particles in a liquid and the particle has a Brownian movement, and also it's a laser that collapses to the particle and produce a diffraction of the light. So there is a formula that measures the diffraction of the light and measure also the Brownian movement of the particle, the nanoparticle, and with that, the formulation gives you the number and the size, yes, of the particle. That is how that works. And give you this histogram, that is concentration and size. I wanted to bring, and I forgot, to bring also one that is before and after purification, that you can see, you can show on around 500 or more. If before the filtration, you can see also peaks in that in 500s or more bigger things, because that was before the filtration, yeah, before the purification. When I say purification, we take out of this histogram all those vesicles and particles or whatever are there that are bigger than 200 and also less than 500. Yes, sorry, 50 nanometer. Yes, 500 nanometer, 50. Yes, and are in more or less in from 50 to 200 nanometers, and this in that peak. So all your samples are in that peak. Yes, particle metrics NTA, Marber NTA, exactly. But those are all NTA, nanoparticle analysis. I'm answering Chaitanya. Yes, thank you. Thank you, Chaitanya. Yes, well, it depends. I was using, she said that particle metric is better. I don't, both are good. Yes, it depends what you use in your, what the most, also that is for the standardization. It's in relation of your hands. Yes, the best that works in your hand when you test. This is for laboratories that testing. The best that works in your hands is the best equipment that you have. If you are not comfortable with the nanosite, it's like that. But I have also my own, yeah. But yes, we can show this with also particle metrics equipment. So in this, when we purify, we have this peak that we have, what we have here. They have different kind of vesicles that are, yes, in different, yes, we have a peak in around 100. They have also in around 200, and all that in the red one. The red, the difference in between these two histograms is that the red one is the media of all the measurements that are in the other histogram. The red is the media of all the measurements.
Are you talking about on the one on the left or on the right?
And the red histogram is in
Okay, this one.
that one.
Okay.
That is the media. Is the media of all the measurements. Yes, is understand? It's the same thing, but the one that have all that colorful lines, each line is one measurement. So the green is one measurement. The red is one measurement. The blue, turquoise is another measurement. If you make the media, yes, statistically, the media of those three measurements, you obtain the red one. Yeah, it's understood. Yeah. So it's the same information. Now is the individual measurement, and the red is the media of all the measurements. Yes, all the population. So they have different kind of vesicles there. They have different part. I don't say part. Not same vesicle. Particles. Just particles. It's what they say there. Concentration particle per milliliter. And anywhere say vesicle or exosome. Yeah.
So then are they contradicting themselves here by putting concentration particles here and then putting exosome count?
Absolutely. I want to know from where they took this exosome count. From where? From where?
They did magic.
Yeah. Yeah. Woo! Yeah. Those are
Okay.
just particles, and they have particles that are in the range of 200 and 150, and a little bit that are higher, very high, that could be maybe, as I say, aggregate. I would like to know if I receive, for example, something. This is educational. For example, if I receive this in the certificate of analysis, an histogram like this, but I have some, yes, like this, for example, and this is a good example. I know how to figure out. Maybe in 300, or if that little peak that is in 300 appear in, I don't know what, say, maybe 600. Well, okay, bigger. I will say, well, those could be, and they say that they purify very well, yes, with a good system, yes, those particles with filters that maintain, yes, the sample in that 50, 150 or 200 nanometers. But in the histograms appear a peak in 600, 500. I say, okay, you have a problem with aggregates. Your vesicle start to aggregate, or you measure without, yes, resuspend, yes, the sample. So all that information you can obtain from the histogram, yeah? When they are aggregates, when you don't purify well. Here they have mix of vesicles and high, because they have three peaks, three different kind of, well, I say vesicle, again, that is particles. I suppose that they purify and they have vesicles in there. But I'm not sure until they show me the other analysis, which are, there is not there. Yes, not there. CD9, well, yeah, CD81, yeah, but
So this seems like it's an autologous blood.
yeah,
Is this what it is?
that is count, yeah, and but independently of that, that is another talk also. Also, we can talk sometime related to platelets, PRP, and autologous that people are using right now. But the main point is that to name that exosomes and say that concentration,
This one right here.
run away.
Okay.
At least ask the company, okay, show me the markers that you use to say that those are exosomes.
Okay. So good. Is there anything else that you want to go over on this chart
It's more
specifically?
to show what they can have, and yeah, and that what they can have when the certificate of analysis come, because that answer the main question of this vocal, which is what is in the
The
vial.
vial.
The only thing, the vial, sorry, my pronunciation.
Okay.
So the main point is like um is like um first step, the only thing that you have to really know something about what is in the vial is the certificate of analysis. Yes, it's the first step. But if the certificate of analysis is incomplete, okay, you have different ways. One is to contact the company, ask for more information. The other is check totally another product that have a better certificate of analysis. And the other, if you have the time and the money, is to, and you have, for example, 3 different samples and you don't know which is the proper one to use and you are very confused, is to just to go to a third party laboratory and say, okay, could you text me all these 3 products and tell me which one, yes, I have the proper vesicles and characterize them. And you have the different information for each.
Got it. So essentially, right, like the way that I've seen it, I mean, if would you consider a red flag if, like, let's say someone calls a company and asks them these questions and they're like, they don't want to show,
The,
or they're not very transparent when it comes to that? I mean, that's probably a huge red flag to where you should be walking out
I was in that situation, and most of the time,
in the situation where you were calling companies and asking them questions.
the person that is on the phone doesn't have the knowledge.
Yeah. Oh, they're probably salespeople, right? They're
Or
just
the
trying to make
information.
sales.
They could know more in relation of the characteristic of the product itself in general. And yes, the other thing, yeah, to talk later is in relation when they characterize the raw material or the final product.
Mm.
So they know very few about all this what we discuss here. Yes, if you ask the person on the phone, do you know the serum that they use to grow the cell? It's like they do.
Well, that would probably be a question for like someone in their R&D department or the scientist that is, behind it. But I would probably say that the person picking up the phone when you first call is, the front desk or a salesperson or something like that. So they probably wouldn't even know.
But that's, I mean, and they don't have to. Yes, they are doing their work in another way. But in relation of this specifically, that is, well, something that is not in the certificate of analysis, and ask the company, okay, you don't have this information, but you really tested. That information is for other people in the company. Some company will, yes, answer why if they yes or not do that analysis, but it's something that, yeah, not all the company will have the capacity to, yes, to tell you. Yeah, and also, well, if there is some company that are growing the cell with this woman fetal serum, is also, I want to talk with that scientist.
Yeah, I want to talk to them. I want to ask specific questions about all
Yeah.
that. Okay. Um, I know we covered a little bit about like which answers are red flags, specifically when physicians are calling these companies, right? Like, what should make a doctor or a clinician using these products walk away on the spot when they're talking to? Um,
Wait, yes. I no first before to finish, and I will ask that is something that I bring before. Is like also another question to ask, if this is important. Yes, this is important, and also related with the certificate of analysis. If If a company present a certificate of analysis and show you, oh my gosh, amazing, yes, an amazing
Documenting.
yes, of analysis with all the characterization and everything. The other thing is this characterization was made in the raw product.
Oh,
So
okay.
immediately when the vesicles were obtained from the production process, or where before you add all the formulation that these products finally had, yes, in conservation or whatever other ingredient that you add to that was after or before to the final product was made. The good answer is to say that was, yes, after
And the final product right before shipping it to them.
product. Yes, because that shows that all the characteristic of that vesicles is maintained, yes, in the final product, in the real product that the patient, yes, that the physician will apply to the patient. Yes, that is where they have to do this characterization.
Gotcha. So you're saying characterize and test and all of that, the final product after it's been in the lab. Does that, by doing that though, like is that going to cost like a lot of, is that going to carry like a lot of operational costs? Because so for example, like if you're doing the final product, right? You're testing the final product, they send the final product to a lab to get that tested. They should obviously be able to know how to handle it, because then also the viability, depending on how they handle it, would that cause any problems with like the viability or anything along those lines
Well,
when you do the final product?
exactly. You are making all the characterization process in the final product to answer exclusively that question. So how to answer the question that you bring here? The integrity is okay, the stability is okay, the vesicles are okay, and the functionality is okay. You are answering those questions when you test the vesicles in the final product. Because if you, they say describing is exactly how biologists will be tested. Exactly, yes, in the final container. Because if you give the imagine that you give this to the physician has this sample with a certificate of analysis that say that shows an amazing, beautiful, as I say, beautiful characterization, all the 3 steps, everything. That was previously to add all the things, and there is something that could, how can I say, that could, well, of course, I hope that is nothing directly not safety for the patient. Yes, all that they add that is safety for the patient. But apart from that, is that really protect the vesicle, for the vesicle to make their function, and the acceptor cells of that patient where that product is applied. You know what I mean? That question is in the final product,
Okay.
not in the raw. You can say, oh yes, I produce amazing vesicles. And after that, you start to add a lot of ingredients, and all your money that you made to make an amazing production laboratory go away, because after that,
Makes sense.
the integrity of the vesicles are not anymore there, and the functionality no. So you
I
are
mean, it sounds like that's how it should be tested.
exactly.
You know, and so
It's
so
logical.
what so why do you think that companies are not testing the final product? Because it's just BS products or it costs money? Is it more expensive than testing it
Well,
in the
you
final
see,
product?
it's not just that. That is also, I want to believe that is lack of education in this field.
Mm,
Yeah. What? Because happened to me talking, yes, inside more, yes, in the cosmetics, yes, that in that field they treat the exosome as an extra ingredient. If you imagine, yes, you don't know nothing about this field of extracellular vesicles and what they do, and know that they are, as I say, active agents, yes, that produce something. They are not a molecule. They are a wild thing with lots of molecules on it that do something. They do something on the receptor cell. So, but they don't see it. Most of them have that confusion that they don't see the vesicles in this way. They see the vesicles as an extra ingredient. So, okay, they produce. They check that they have the ingredients and they add to the formulation. That's for a drug or for whatever other components part of the formulation. Okay, that is, yeah, okay, that is good. So the way to proceed, and you know that all the formulation all together, you try, yes, and you have that it's effective, yes, you try it's effective, perfect, and the potency, whatever. But this is not that way. This is not another ingredient. Vesicles are not another ingredient.
Yeah.
They have to be really maintain the integrity of the membrane. Also, if you go further, the corona, because whatever ingredient that you put in, change the corona, the things that are going to be attached to the vesicle to be recognized to the receptor cell, that reduce the potency. Yeah. So and the possibility for that agent to reprogram the receptor cell. Yeah. So it's not another component of the ingredient. That is the problem. For that reason, because they see this as another ingredient, is for that reason that they don't characterize or recharacterize in the final product. I know that what the companies do is that when they finish, before the final products, when they finish just to purify the exosome, to produce the exosome, to purify the exosome, they have to check that they have the exosome in the proper size. Yeah, size, that they do the purification in a proper way. So they check that. They must check that. But after that, they start to put all the other components that are part of the product. But they don't recheck that. They don't recharacterize to say if everything is still good there.
Okay. Okay. So
And of course,
what?
they have to do that with every batch.
Yeah, every
But
batch, and that can get expensive, right? But I mean, if they're in business to do that, I mean, they need to cost, right? That they need to factor those costs. Because at the end, it's patient safety. If we
as
don't,
I
if
said,
we don't,
from one way, we have to start. But we know what we have. The best the way to go and to improve in this field is to do the correct thing with what we have. That's what I mean.
so one thing is we went over vetting the vendors, how to read a COA, what to look for, and obviously, I think you did a phenomenal job with all the questions from everyone. And beyond,
For the question.
I'm sorry.
No, I say thank you for the questions. I love that.
No, yeah,
It
we've
to
been
be
getting some
clear
great questions
to
from the
everybody.
audience too.
Yes, I
So
love
I appreciate that as well.
I
So
love that is yeah.
yeah, it's great. So we need more scientists, by the way. Anyone that's hearing out there, I would like to kind of do more of these and then have, I would love to do scientist to scientist talk because obviously I don't know. I'm not a scientist, right? So having scientists talk is I think is
Yeah.
good, right?
Very good.
Sharing knowledge and all that. But anyway, okay. So let's go back to like a physician, right? So obviously, beyond picking a good vendor, that's one thing, right? So there's a lot of that goes into it in how to pick a good vendor. If I'm a doctor, what do you recommend for me to protect myself? Like what documentation and my due diligence? So like do your own research, ask the questions like you mentioned, right? Uh, First of all, like what are we going to be uh, I don't like using the word treating because of the FDA, right? But what are we going to use it for, right? That's more of a safer thing, right? I would say. Because I know treat, cure, those are very trigger words by the FDA. And because the exosomes are not FDA approved with any indication as of right now, I
Yes.
don't like using that. But
Yes. Well, yes, finish and I
yeah.
will.
Well, yeah. So all I was going to say is that I don't want to use those words, but like so the first question is, what are you going to use it for? From one to what are you going to use it for? What is the source? And that's the safety, right? So what should a physician be and documenting when they're using these exosomes? For example, like, okay, saving all the certificate of analysis, documenting, tracking the patient, any adverse
So
reactions, effects,
exactly
right? Like,
that.
what are the things that they
Exactly.
should be
Or
doing,
yeah.
not just from vetting? Okay, so
I will say the best way to, well, they 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 know very well why and how they run their own clinic. Yeah. I'm not here to tell anyone how they have to run their own clinic. That's the start from there. I can help to understand, yes, what they how
well, you are right. I feel like part of that as well. Some of them, I mean, let's be real, right? I believe doctors, they went to med school not to become business people, right? And
yeah, exactly.
there are some
And
doctors that are smart in business, right?
trying
But
maybe indeed, yes, knowing
you
in
know,
this kind of
exactly. So it's not the way I see it, it's not their fault that they don't know
yeah.
how to run a business. Most of them. I'm not gonna say all of them, but most of them maybe they were not taught on how to run a business, right? So
Apart from the business, I say in relation of the patient and how they do
yeah,
that, but in relation of the vesicles and how to, I may say how to go to really for the clinic to have and to work with a good vesicles is to start from the beginning and really choose and first to do the research and really check what is in the market. Yeah. How they sell that with what we learned today, if they follow all those steps, yes, if they have all the characterization to read the certificate of analysis, and after that to choose, yes, what is, and when they choose, maybe from the certificate of analysis, they start to do the research and say, okay, I want to know more about the science behind and the production part behind this science, this sample that I want to use in my clinic. To maybe try to find the correct person that answer those specific question of how that product was made. Yes, like the filtration, and let's say the how the cells were growing, all those things. If they answer that question, great. If they not, and you really want to work with, yes, with that samples, or you really want to know what is in there, you can go and test. In relation of the patient, of course, I say that is amazing to start with a product that you did all the work, yes, all the research about that product, and you start to apply um, in your patient, also following as the regulations
of
and
course.
and also say, well, and after that, of course, that would be amazing for the clinic if for that specific product that you did all that research and know that, and you know that is good, how that works on the patient. Yes. Apart, if you have to go further and to have, yes, if to check the integrity and to check that is part of the characterization, but if you want to go further and to check the potency of that specific sample, if product, if the company don't provide that, so you can do it, but also in your patient and to check, yes, in the long term. That is what the vesicles do, because if it's in the short, if there are not vesicles there, most of the results that you have is short term. Is as much as you apply that, you are going to see the benefits. But with the vesicles, because they reprogram the cell, that is the greatness of the vesicles, yes, and most the exosomes are reprogrammed receptor cell. When I say that, this is long term. So you stop applying and you still have the result. So if you check, that is to check on the patient that the vesicles and the product that you choose, yes, and you did all the research on is good on your patient, and also to check the side effect that could have, yes, the side effects will be more in relation.
The side effects, right?
Yes,
This
not the side effect. Not for the not most for the vesicles itself. I think it's more two things. One it will be more for and the formulation, and I say most that come from the formulation. Whatever, yes, I don't know, condition that you start to see in your patient, and the other is in relation of the sterility, maybe, yes.
the sterility.
That is also when people ask me, okay, how much time the vesicles could be? I say, it's not because it's not it's not so much related with the vesicles and the exosome because the membrane is strong if you keep outside, but it's more a relation of how much time that will still sterile, yes, outside is is that.
Okay. So obviously we know that, the FDA is there's no FDA approved exosomes for whatever reason. Like what besides that, is there anything else that physicians and clinicians should be aware on the regulatory aspect? I know you mentioned a little bit about topical and things like that, but this is not really a regulatory topic, but it's something good to understand the regulatory aspect of exosomes, right?
Yes, it's more because most of the I will add that most of the well a bunch of I'll say most a bunch of for example cosmetic or regenerative they are also doing some studies that go through the therapeutics and they are starting so they have some projects there that are running so those have all this yes process made so maybe and go to those companies that are doing the correct thing in that way. So those are products that are more how can I say more trusted because they are in that line of the way. The other is well, and to ask, yes, and to ask if they are in that process because that's also make things easy, yes. For the regenerative and for the cosmetic to use this product. But you have yes to be very very careful how you use them.
Uh,
Be very
from
careful. And
from your experience, I have a question. From your experience on the regulatory side, a little bit, I've realized that a lot of physicians, clinicians, and people don't have never heard of the expanded access program or the right to try. Right? These are pathways put by the FDA to use unapproved drugs for there's certain criterias, right? There are two different ones. Each state has a different right to try. And do you are you familiar with say like with any clinical trials or anything like that? You or any doctor using the right to try?
there are
There are? Okay,
yes,
cool. And and I feel like a lot of people, one, they don't they maybe they've never heard of these pathways, right? That is put by the FDA, but they're out there to actually help patients, right?
yes. But for the
Um,
reason I say the product have been in some way investigated. Yes, that is the main step. So the the product that you will use has to be in some way right now in the process. Yes, in a trial or whatever that are part of the VA. So they have more. You protect more in that way when you use the right to try.
yeah. Okay. Yeah,
For a
there's certain
product that comes
requirements.
from nowhere and the FDA have no any clue about what is that and that is not the way to go. But if they are a part of some yes investigation, that is the way to go. Yeah, the right to try is yeah, is something that you can go for. Well, also for those people that really that the right to try that really need that because they don't have another choice.
Another alternative, yeah. Um, then then I know there's well,
I know companies that they are using right
if
now.
I'm not mistaken, I believe the right to try essentially it has to go it has to be in place in phase 2 clinical trials, I believe, in order for you to use that, right?
And there
And
are
then yeah, there are exosome companies that are in phase 2, phase 3.
yes, and there are also there are, I don't know how they do, but I know also companies that are not in that, and they are doing the right to try. I don't know, and that's how they process that.
Yeah.
That have to be that you have to talk with them on regulatory
Yeah,
person.
I did a little bit of research on that,
Yeah,
and honestly, if you just Google expanded access program,
yeah,
the
exactly.
FDA has a
Yeah,
very good information to where it has all the, frequently asked questions.
of course.
Um, there's I believe, yeah, there's a form that you can fill out as a
Oh,
physician, and there's
as
different
a
types of.
physician, yes,
Mhm.
and the patient have to sign that.
Yup, yup, and then but so it's very
That's
informative, I believe.
protect the physician.
Correct, correct. Protect the physician. There's different. There's a lot of criteria and certain requirements that are needed, and the right to try is a little bit different. So those are two different pathways that I always like to mention because
Yeah.
when I'm in conversations with physicians or people in the industry, they've never heard of it, and it's wild to me to where like I'm like, you've never heard of that? That's
Yeah,
crazy. Like
yeah.
look it up. And then I feel like, in my opinion, the people that have heard of these pathways, they're kind of lazy to fill out all the paperwork, and they don't even want to do it.
That is a pity for people that really need it. Yes, because there are people with very very terrible illness
Yeah,
that
yeah.
they can
I have
benefit
no other anything else, and rather to try
from
that.
the work. Yeah,
Yeah.
and
Okay.
yes, they can
So
benefit for that reason. Again, the future of medicine is vesicular.
yeah, yeah.
So they can do
So
that, but for do that we have to protect the vesicle and make the vesicle do the work. Yes, once they
yeah,
we really protect them.
yeah. Well, and then that from there, yeah. Once you treat the patient using those pathways, and or the biggest thing is data integrity, right? And making sure that you're keeping track of that, you're following the patient, which is huge. So I understand it's a lot of paperwork, but I mean, physicians signed up to be physicians, and they should be keeping track of all of that, right? I mean, you know, it's a lot of paperwork, I know, but
Yeah.
so
We
in
do
order.
all the hard work also to characterize, yes, the
Well,
best.
that's true. Yep,
That is
that's
a
true. I'm
process.
sorry. I'm not taking away. You guys do put a lot of work in the laboratory. I'm not trying to take that away from you guys. so, so
So
just to kind of,
everyone do they work, yeah.
yeah. So just to kind of like recap a little bit, there's a couple of more questions that I have here. So like from a doctor, obviously we went what 2.5 hours, and what is one thing that you want a physician to take away from this conversation? What is that one thing that you want them to take away?
Well, that comes from the title of this postcard is don't believe in what is it what is the just the vial say. Yes, yes. The main point is now you know what is needed, the questions that you have to ask, and the research that you have to do by themselves now, with the conditions that we have now, to really have the best products in the vial that you are going to apply in your patient.
When you say best, would you
The best
probably
possible.
mean like the safest,
What I
or
mean, yes, exactly. What I mean is in relation of the safety, in relation of what is there to really know. It's not just okay. What is here? And ask what is they say exosome, trillion of exosomes. When they say trillion of exosomes, I would say, what? What you
yeah,
are telling me? And after that, dig more deep and
yeah,
discover what is there. Try
ask
to
questions.
find, yes, try to now they have the question to do. Yes, now they know the question that they have to make.
Ask.
So yes, to ask. So the is to ask those questions and know very well the product. Yes, that
Yeah,
they have in the clinic.
yeah,
That is
and
possible. It's not.
yeah,
And if not, is to ask start to ask the companies that come to you and the vendors say, okay, tell your company that please do this in this way because we need as a physician. This is the main thing to help. If they want to help, what I ask them is to, if a vendor or if I contact with the specific companies, production companies, or product company say, okay, we physicians, we need this information to really know what we are applying to our patients. So please, could you make the work and give us this answer in the certificate of analysis, or when we ask and call you, and we need this information.
Yeah.
We need this information.
And also, I think, in my opinion, I believe if you are a manufacturer, if you're producing these exosomes, or particles or extracellular vesicles, you should expect people to ask these questions, right? So, like, for example, I think that transparency, any company that's very transparent with whatever they do without maybe, let's say, giving their secret sauce or the proprietary process
And
or
also,
whatever, right?
and also, they will be, if they do the correct thing, they will be very proud to share that.
Yeah, exactly. So I believe that, like, if I were a manufacturer and I have scientists and people asking me these questions, I'll be an open book. I'm like, here's everything. Like, this is what we do. This is all of that, right? Because I know that that will build trust. And if you build trust, people will, I guess, buy from you, right? And then, everything goes along, right? Goes from
Yeah,
there.
but the problem with this is also finishing in how to reduce cost. Yes,
Follow the money. Everything is about the
I
money, right?
I understand that. I understand that. But you can reduce costs doing the correct thing. It's again, we have what we have. There is a way to do the things correctly and not to start to, I don't know, yeah, not give the proper information or start to reduce things that are, if you say that you have exosomes, you must show that are exosomes there. And if
Yeah,
you sell
of course.
media, yes, instead of purify them to reduce costs, tell what is in the media and also tell that this is risky.
Yeah. Okay. So what gives you optimism that the field is heading towards standards and improving in exosomes? What gives you optimism?
Well, is what's in all is this again. The future of medicine is vesicular. After all 20 plus years study these vesicles, um, I know that the potency that they have not is in, as I say, in cosmetic, in therapeutics, in diagnostics. Oh my gosh, in diagnostics, in to very very early detection of cancer, for example, and in now also in drug delivery, all that um and also the results, yes, that we start to having on people with really in most in the studies that we have in therapeutics, yes, and the results of that, and also now more and more the physician and in general the community want to learn more, yes, about what is an exosome, what is this, how this work, how this, and all the variety of that is for another podcast, the plants, the ones that come from the bacteria, all that thing. So it's coming more and more. I'm really happy that we have that now. Yes, that the first jump. We are in the first jump from all the research that we have in the in the in the in the academia. Yes, in the science, jump to the industry. This is the first jump. Now we have to put things in order there. Yes. So for that reason, I trust on this, because now is the time to start to put things in order and to know that, okay, the cost is important for the industry. Now is the scaling up. Yes. From what we do in the scientific world to now the industry, all the scaling up, all the standards that we want to add there, all the reproducibility, all the things. Now, well, little by little, we have to work that. And I'm giving also all myself to this now that I'm, yes, I'm continue, yes, doing things in the academia, continue in contact and collaborating with Boston University, like in college and other laboratory, but now I'm more to solve this kind of problems that I know that I can solve it from my years and in this and my knowledge on the industry in this
Okay.
field. So I'm doing things right now. I say right now, as we start talking, it's not to be patient that things will improve. That is, or be confident that still can improve, but doing things. I'm a doer, so doing things. What I can do to improve that, and from the part of physicians that are here, and for people that produce vesicles, is that with what we have, what is the best thing that I can do to not complicate? Yes, now, because sometimes companies also start to confuse ones or start to, I don't know, to name things differently or start to put information that is extra then and people start to become so confused. And I hear also my friends that say, oh my gosh, I don't understand nothing. I don't want nothing to do with exosome. I don't understand that and
Yeah.
that. And I say, why?
Yeah. I have a, it feels like a lot of people are, unfortunately, it's sad to say this, but I feel like a lot of people are tired of exosomes, like because they've,
That
it's
that is a
and
pity.
it,
That is
yeah,
that is the way that I'm on. Yes, it's a pity if that was that way because what's happened, and I started to hear that what's happened is because the people on the street start to say that. After that, the company will start to be aware of that. Why we are producing exosome effects if the people start to not be interested. Also, the investors start to to lose interest and to get funding for that. After that, and also the science will be affected because after that the agencies that give the grants to study these vesicles will be reduced because there is less and less interest on it. After that, the amazing work that scientists are doing in this field also will be reduced with something that is really good. Could be that the future of the medicine will be the future of the medicine if we do the correct thing right now. That is.
beautiful, beautiful, beautiful. So, um, last question, Dr. Marzell. So, where can physicians or scientists continue learning more about what you're doing, and how to source responsibly? Sorry,
Well, I'm most on LinkedIn and also
LinkedIn.
yes, and also I'm starting different ways to give education. So I really want to know the people that are interested on education to start a kind of program related with education in this field. But for other part, I'm also starting to create this safe that, as I say, we start to have laboratories around the world. Now we have around 8, 9 laboratories, and we are signing agreements. So it's a reality. We are doing also we are building some platform with this all related with blockchains and also in crypto and all those things where we collect all the data from all those different laboratories, and this laboratory will give a good certificate, good, not well, characterization or bad, and where is the risk. Is not to put the point on the companies that don't, I don't know, the characterization was wrong. It's just to help and to say, well, you can improve this. This test was not so good, and I know why, because most of the time I know why that in which is exactly step things are going wrong. So it's just to say, okay, I can give you the advice on how to improve that, and also later on, as I say, is to certify the process and to have an amazing standard finally, yes, to go to the FDA and to make this a reality. So if you want to help with that, it's just to, well, start to tell the companies that they must test, they must to characterize this, they must to go to companies, and I do that in those laboratories, the 3-party testing, yeah, to me or whatever other third-party testing laboratory that is totally independent of production and do this characterization properly for the physician to have all the information and to put as much information. You have to be proud if you do the correct thing and you know that you have the best vesicles in your
In the
field.
laboratory, in your field.
Yes, if you're producing, you have
I'll
to
say
be,
that loud and clear.
yeah,
But
exactly.
and with proof though, because the problem is that everyone is saying that they have the best exosomes, right? But according to
Okay,
who? And right?
exactly, but prove that you have at least vesicles still in your final product. Yes, in the product that the patient, yes, that the physician will apply to the patient. So that is the main way to go. I'm totally, if we do that this way, wow, as I say, the future of medicine will be fantastic and will be vesicular, really vesicular. And because what they can do is just amazing.
Amazing things, huh? Okay,
Mhm.
awesome. So that pretty much finalizes all my questions that I had for you here. Is there anything that you want to leave, the last words that you want to say to the audience?
Well, I prepared a kind of guidelines and that we are going to prepare the you remember the guidelines to get them at some point with questions and that.
You sent it to me.
Yeah, long time ago. You remember the guidelines, the
Do you have it easily?
well,
Was it the checklist?
yes, the checklist.
Okay, so I
On the
couldn't find it, but if you can send it to me, that'll be
you
okay,
can,
and I can show it to. Yep, and I can show it. So
and the other is, well, we can prepare that the best certificate of analysis sample.
that's a good one.
That is good to have.
That is a good
And
one.
again, whatever question, whatever, I love to discuss all things related, yes, vesicles and discuss science, and yes, I'm totally open to that. So you can reach me most, yes, I would say by LinkedIn. Little by little, I'm start to, yes, to post most and most educational parts and different kind. Also, I will start also to talk about most regulatory and also some news or stories. Yeah, some Fridays I want to publish some stories from different companies. What's happened with this and that, what they do, what's happened. I don't know.
Yeah, like
And
just kind of like behind the
so
scenes. Are you working on sending me that checklist that you're talking about?
sorry.
Are you working on sending me that checklist that you were talking about?
Yeah, I will send you if you want. I
Do
will.
you? I guess my question is, do you want to cover it now on the podcast
No,
or?
we will send it because I think we
Oh,
covered all the things that is in that.
I see.
So it's for them to have it.
I understand. Okay, good. Okay, great.
We
So
can
I
send that to them later.
okay, I get it. So like I guess for anyone that is listening, if you it's a check, can you go over at least like what the checklist is about? Because honestly, I forgot, Dr. Mazzeo, to be honest.
Well, it's all about what we were talking in relation of the question that they have they
For the vendor, like essentially
made
like what's okay.
on how to know the question that you have to make to
To ask the vendor.
the vendor or the manufacturing, and also the question that you have to make yourself. Yeah,
Okay,
in relation of okay, what I have to what is needed for me to know about what is in the bill.
got it. Okay,
All that.
got it. Okay. So a checklist on how to vet your
Yes,
vendor, pretty
I
much
go through
essentially
the
everything we
certificate
went through today.
of analysis, what must to be there, the question, and yeah,
Okay,
and how to identify, yes, all that.
awesome. Yeah.
And I'm open also to discuss that, so people can
Okay.
reach me. I'm very, yes.
So for everyone that is joining, I will share the list, all your emails with Dr. Mazzeo. So what I would say is probably expect a email from her directly, kind of introducing herself or whatever all that, and then obviously you guys know how to reach me as well. And I believe if anyone is out there listening, because I usually post these on my YouTube channel as well. So if you're listening on YouTube now, you can check all the links in the description below. And then also, if you're a scientist, you want to get into these conversations, like let's say I can host Dr. Mazzeo and have two scientists or more. We would love that because we're trying to just literally create a community with
You could
education and all
set
that.
up to make a podcast with another scientist.
Yeah, with other scientists. Yeah, and then just kind of because I feel like if we were to bring another scientist,
Yeah, that would be
to the part, the value would be even higher, right? Because
yeah.
you guys can go back
Oh,
and
no.
forth. And you guys can all geek out, right, and nerd out or whatever with all your questions.
Yeah,
So,
that would be good. That would be good. Yeah.
so all right. So, well, if there's any last words, Dr. Mazzeo, that you want to give before we
I
bounce.
think I cover most of the things that I want, yes, to cover. Yeah. And
Okay.
yes, we can also later on. I want to talk also about, as I say, the vesicles. That's hard for another podcast. Maybe exosomes about what happened with exosome or vesicles that come from plants. Well, vesicles
Yeah.
more from plants.
Now I've been seeing a
And
lot of.
also the autologous
I
thing.
was gonna say that there's a lot of now autologous from PRP from
Yes, because
blood.
that come that come also from the regulatory. So it's start to go for the regulatory. Yes, the autologous is something that you can in some way manage there. Regulatory from
That
the
makes sense. That makes sense of why they are trying to
but
go around, right?
it's also a good things to talk about the difference that an autologous exosome, exclusively exosome, an autologous exosome in relation with uh, an exosome that come from, yes,
Neonatal.
it's the neonatal.
But
Yeah,
then we all go back to like, okay, it's an autologous exosome, but where is the source coming? Where is this exosome coming from, right? It's autologous, but from where in our body,
where in
correct?
your body, exactly. Mm-hmm.
So that's a different conversation. That's a crazy talk. Okay, cool,
After the
awesome.
PUP, the platelets,
Yeah.
all the things that they say, they talk about the exosomes and yes, what they obtain after they activate the platelets. That is another conversation.
All right, cool. Well, I'm looking forward to that. Well, okay, cool. So everyone, happy Friday. Thank you guys
Happy
for
Friday. I
being
love
here.
Fridays.
Yep. Thank you for being here almost 3 hours. I appreciate, and I apologize about earlier today with all that banging, but we got
Yes,
to figure it out.
always.
So thank you guys for speaking up because I was hearing it, but I was kind of like, maybe the
Yeah.
noise cancellation was probably like working or whatever, but I guess it wasn't. So,
Yeah.
but anyway,
Happy birthday.
yeah, it is my birthday. Thank you so much, Dr. Mazzeo. I'm going to celebrate now with my family. We're going to have some fun. I'm going to spend some time with the kids.
That's good.
So everyone, thank you so much for joining us, and we look forward to doing it again next time. And we will be doing more of these for sure. All
Yeah,
right.
sure.
All right, guys. Adios, adios, adios. Thank you guys so much.
Ciao. Ci vediamo. Bye.